Good afternoon, guys. I hope I am audible and my screen is also visible to you guys. So, I am Rishabh. I am from IIT Kanpur, and today we'll be discussing about the discussion week 5 of the course Introduction to Cell Biology. In this video, we'll be discussing about the cell wall, cell membrane, and the starting of the cytoskeleton. Okay, sorry. So, let's move forward. So starting with the cell wall. So basically cell wall or the cell envelope is defined as the cell membrane sorry. So there is a term called as cell envelope. So basically cell envelope is the covering of the cell. Okay. So it will involve both cell membrane as well as cell wall. Okay, and if any other membrane that is present outside of these two membranes. Okay, in case of bacterial and plant cells we have a specialized cell uh, membrane, that is cell wall. Okay, so it is a rigid kind of structure. Okay, it's tough and rigid, and it is actually surrounded or located outside the cell membrane. Okay, and it helps in providing the structural support and protection to the cell. Okay, so the basic thing to remember about the cell wall is that it is rigid and unbreakable. Okay, it cannot be broken down easily. Specifically, they require certain enzymes to break them. Okay, for example, for bacterial cell wall, that is made up of peptidoglycan, lysozyme is required to break that down. Okay, so likewise, these cell walls are like difficult to break. Okay, rather than unbreakable, they are difficult to break, and they require specialized techniques or enzymes to break them. Okay, that is how they provide structural support and protection to the any kind of cell in which it's present. Okay, this was the basic introduction about the cell wall, and as you can see here, this orange colored region is basically the cell wall in case of the bacterial cell, and this green colored region, so this green colored region that is here, this is the cell wall in case of the plant cell. In case of the plant cell, it is basically made up of cellulose and hemicellulose, followed by certain modifications like polysaccharides and proteins. Okay, so this is made up of cellulose and hemicellulose, and in some places that is where the secondary cell wall is there, that is made up of lignin in case of plant cell. Okay, and this, because plant cell are also dividing in nature, so there are two kinds of cell wall in that case, that is primary and secondary. So primary cell wall is basically smooth. Okay, because these are present in those cells that are continuously dividing. Okay, so if it would be difficult to break a cell wall during division, then it would be highly energy requiring process. So that's why in the primary cells the cell wall is present, but that is most probably thinner, so that not much energy is required to break that particular cell wall. Okay, in case of secondary cells, what happens? The cell wall is rigid. Okay, in that case, the if we have to break the secondary cell wall, we have to require more energy to break it down. So that's why it contains molecules such as lignin that are difficult to break. Okay, and that are present in differentiated cells that do not divide. Okay, so this was the basic introduction about cell wall. If you guys have any doubt, you can ask. So let's move on to the questions. The first question is the composition of peptidoglycan layer is. So what can be the correct answer? Okay. Any more answers? Okay. So the correct answer is option 2, that is N-acetyl glucosamine and N-acetyl muramic acid along with certain amino acids. Okay. So this is the basic monomer for the peptidoglycan. Okay. So it's a heteropolysaccharide basically, and it has two monomers linked by a glycosidic bond. So this is N-acetyl glucosamine and this is N-acetyl muramic acid. Okay, so these both are linked by the glycosidic bond, and what happens, there is a pentapeptide also that is present attached to the N-acetyl muramic acid. Okay, this list of the pentapeptide has different amino acids in gram positive and gram negative bacteria. Okay, so the amino acids that are mentioned here, it's actually a pentapeptide. So this is present in a chain kind of structure. For example, this is the chain of the heteropolysaccharide, the NAM and NAG polymer, and this is the second chain, and the amino acids are present like this, forming a mesh kind of structure and making the cell wall more rigid. Okay, so this is how these amino acids are present in the cell wall. Okay, so any doubts in this question? Okay, so let's move on to the next question. The function of alcohol in a gram staining protocol is. So what can be the correct answer? Okay. Okay. Any more answers? Okay, so the correct answer is option 2. That means it dissolves the outer membrane of a gram negative bacteria. Okay, so gram staining is a procedure that was uh, devised by Sir Gram. Okay, and what he did was he was basically tried to like differentiate different kinds of bacteria. Okay, so there are two kinds of bacteria on this particular after this particular staining. So there are two kinds of bacteria: gram positive and gram negative. Okay, gram positive bacteria are those that retains gram stain. Okay, and gram negative are those that do not retain gram stain. Okay, how? We'll see the procedure first. So what we generally do in our lab is we take a smear. For example, you have curd. You would have done this experiment in your labs also. You take a slide. You take one drop of curd here, and you take another slide. tilt it a bit, and then you slide it down through the whole slide. Okay, so this is basically creating a smear. Then what you do, you give a brief like heat to this particular slide, so that the smear that is there fixes on the slide. Okay, now what you do, you put a stain of the crystal violet, so that is a purple colored stain that stains the cell wall. Okay, so what will happen, the all the bacteria that are present in the particular smear will stain purple. Okay, then what we put a second component, that is iodine. Okay, so iodine is a mordant that basically stabilizes the stain that was given to the cell wall of the bacteria. Okay, so what will happen? The cells that were purple will remain purple, and the purple color would be more stronger in this case, because we have added a stabilizer in the particular smear. Now what we do, we put an alcohol, so that is a decolorizer. So what will happen in case of, if we put the alcohol, the cell membrane that is present, that the membrane that is present outside the cell wall will disappear. Okay, so all the bacteria that have a particular wall or a particular membrane outside the cell wall will actually dissolve because of the alcohol. Okay, so in that case, what will happen now? There would be two kinds of cells. One, the cells that retains the purple color, and the other ones that were colorless. So now to differentiate between the purple colored and the other cells, we stain a co-stain that is saffron. So this is pink in color. Okay, so now the cells that retain the purple color will remain purple, but the cells that were colorless after providing alcohol, then those cells will be now pink colored. Okay, so the purple colored cells that retains this crystal violet color are called as gram positive cells, and the cells that do not retain this purple color and appear as pink after the imaging are called as gram negative cells. Okay, so after this staining, he basically inferred that there are two kinds of cell, two kind of bacteria, that one is gram positive, that have a outer covering of cell wall, and the other one were gram negative, that have another membrane, we call it as an outer membrane, that is covering the cell wall. Okay, so it covers the cell wall. That's why when alcohol was added, this outer membrane was dissolved, and now when we add a co-stain, that is that stains the cell wall of that particular bacteria. Okay, so the function of alcohol here is to dissolve the outer membrane of the gram negative bacteria. Okay, is the procedure clear, and the question also? Okay, if no doubt, then we'll move on to the next question. So the gram positive bacteria have dash peptidoglycan rare. So what can be the correct answer? Okay. So the correct answer is option 3. That means they have a thick peptidoglycan layer. Okay. So this is how a gram positive bacteria is basically represented. So this is the schematic. So this is a peptidoglycan layer, that means the cell wall, and after this we have the cytoplasmic membrane or the cell membrane. Okay, and this is a 3D reconstruction of that particular thing. So this is the purple colored bead is N-acetyl glucosamine, and the light blue color bead is the N-acetyl muramic acid. Okay, and after that we have teichoic acid, that is a modification given to the cell wall for its rigidity, and this orange colored extensions are the amino acids. Okay, so this peptidoglycan layer, as you can see here, is very thick in case of the gram-positive bacteria. That's why these bacteria actually retains the crystal violet stain because of its thickness. Okay, and after this, this is the cell membrane, the phospholipid bilayer that is protected by the cell wall. Okay, here an interesting thing to note is all the bacteria that are actually antibiotic resistant in today's scenario are gram positive, nearly gram positive, because they have a unique feature that they have D amino acids. Okay, and what happens, the more or less all the like in today's universe, the physiological amino acids that are present are L amino acids. Okay, and in case of gram positive bacteria, these D amino acids are present, and this make gram positive bacteria difficult to be difficult for an antibody to be act upon. Okay, so that's the major reason these are there, and this is a electron micrograph of this particular gram positive bacteria, and as you can see, this peptidoglycan layer, this one is really thick, and after this layer we have a small layer that is the cell wall, cell membrane. Okay, so any doubts in this question? Okay, okay, so let's move on to the next question. Periplasmic space is, so what can be the correct answer? Okay, any more answers? Okay, any more answers? Okay, so the correct answer is option 4, that is the space between the outer membrane and the cell membrane. Okay, so this is in terms of the gram negative bacteria basically. So as I told you, in case of gram negative bacteria, what happens is we have a membrane, that is the outer membrane, a thin cell wall, and the cell membrane. Okay, so this outer membrane that is present, the space between the outer membrane and the cell membrane is called as the periplasmic space. Okay, so this is in terms of gram negative bacteria only, because outer membrane is not present in gram positive bacteria. Okay, so this is how a 3D reconstruction of a gram negative bacteria would be seen. Okay, so this is a lipopolysaccharide layer that is called as the outer membrane. This have porin proteins that are pores, so help in transport of the molecules. Okay, and it have extensions. It is basically a lipid bilayer. Okay, that is attached to the cell wall. So this second layer is the cell wall basically. Okay, and then we have the sorry, this is the second layer is the cell wall, and after this we have the cytoplasmic membrane or the cell membrane. So this periplasmic space actually helps actually stores all those enzymes and proteins that are involved in degradation. Okay, so basically those enzymes that have a special capability like of degrading pesticides or special organic molecules have certain enzymes present in this periplasmic space only. Okay, so and this also holds a special path to absorb different ions and different substrate molecules. Okay, and this is the electron micrograph of this particular gram negative bacteria. This is the capsule, that means the outer membrane, sorry, the outer membrane. Then after this is the cytoplasmic membrane, and between them we have the periplasmic space here. Okay, so any doubts in this particular question? Okay, if not, then we'll move on to the next question. Secondary cell wall is present in which of the following organisms? So, what can be the correct answer? Okay, okay. So, yes, the correct answer is option one, plant cells. So, they have a secondary cell wall. Yes. So, like I said before, all the actively dividing cells and the metabolically active cells, like photosynthetic, secretory cells, those have a primary cell wall that is made up of only cellulose, so that the division is not an energetically enhanced process. But those who which needs compressional strength, such as those present in the wood. Okay, so wood part of the plant, they requires another wall. They requires thicker wall called as the secondary wall or the secondary cell wall. Okay, so in that case, we have the uh, this is the multi-layered cell wall that is required by the plant cell to have the grip or the compressional strength. Okay, and it is mostly made up of lignin. Okay, and this is actually multi-layered, and those can be also like lignin as well as cellulose. Okay, so this is the major function of the secondary cell wall that is present in the plants. Okay, and those differentiated tissues that are majorly involved in the mm, transport of the molecules. Okay, so those also have this secondary cell. Okay, so any doubts in this question? Okay, so let's move on to the next question. So now we'll be moving from the cell wall to the cell membrane. Okay, so cell membrane, as we all know, is a phospholipid bilayer, and it is basically described by the fluid mosaic model. Okay, so it is a phospholipid bilayer, and these are basically the outermost covering in the animal cell and the second most covering in the plant cells. And this is a basic feature of this is semi-permeable membrane. Okay, so semi-permeable or selectively permeable means that it only allows the transport of specific molecules. Okay, and it is basically it controls the entry and exit of both solute and solvent. Okay, and we have okay, so the functions are it maintains the integrity of the interior of the cell. Okay, so it basically separates the extracellular fluid from mixing it with the cytoplasm. Okay, so that's how it actually maintains the integrity of the cell by maintaining different organelles inside and protecting it so that it can actually function and produce different kinds of stuff so that the body can function. Okay, so this is the cell membrane, and it actually separates out the extracellular fluid from the cytoplasm. Okay, then it helps in support and maintaining the shape of the cell. Okay, then it regulates cell growth through the balance and of endocytosis and exocytosis. Okay, so what happens, there is term called as cell growth. Okay, so what happens is whenever there is a increased amount of substrates that actually being like accumulated in the cytoplasm, so what happens is this cytoplasm actually grows. Okay, so this cytoplasm growing gives a signal to the cell membrane so that the substrate that are extra for the cell can actually be removed from the cell. Okay, and through the process of exocytosis. Okay, so that means exit from the cell. And if there is shrinkage in the cell, then the cell membrane allows the endocytosis of the substrates or the molecules so that it can actually increase the amount of cell for the optimum conditions. Okay, then it also helps in signaling. Okay, so cell cell communication, if we have anything that have that is disturbing this particular environment inside the cell, then it will actually send the signal so that the cell that are surrounding it can help this particular cell for the stress to be overcome. Okay, then the last point is it is selectively permeable, so it allows only the entry of the specific substances into the cell. Okay, so, one major thing regarding the signaling is I am giving an example of apoptosis. So, that is programmed cell death. So, whenever a cell becomes old or it is diseased, that means it needs to be cleared from the body. So, now what happens is this cell gives a signal, that means how it gives a signal. There are many hallmarks of this. So, one of the hallmarks is the the cell will become more negatively charged as compared to the normal cell. Okay, so this negative charge actually gives the signal to the other immune cells so that they can clear off this particular cell and other cells can divide and replace this cell. Okay, so this is one example how a signaling can actually takes place with the help of the cell membrane. Okay, so this is the basic structure and the functions of the cell membrane, and the fluid mosaic model we'll be discussing in a couple of questions. So, any doubts in this particular slide? So let's move on to the next question. So what is the primary component of the cell membrane in all cells? So what can be the correct answer? Okay, any more answers? Okay. So yes, the correct answer is option 3, that is phospholipids. So the major component of the cell membrane is phospholipids. Okay. Cellulose is a part of plant cell wall. Peptidoglycan is a part of bacterial cell wall, and chitin is a part of the fungal cell wall. Okay. Now, talking about the phospholipids, as we have previously discussed also, they are amphipathic in nature. What is meant by amphipathic? It has both hydrophilic and hydrophobic groups. Okay, so we depict the phospholipid as this part. Okay, so this is the head of the phospholipid, that is a hydrophilic head, and these two are the fatty acid side chains. Okay, so this is fatty acid chain 1, this is fatty acid chain 2. So these two are the fatty acid side chains, and this is the hydrophilic head. And this is how it is arranged in the form of the phospholipid bilayer. Okay, so moving, keeping the head towards the outer side and keeping the hydrophilic side inside, so that they can maintain the hydrophobic nature of the fatty acids, protected from the watery environment. Okay, and this is how they form the lipid bilayer. Okay, so any doubts? Okay, so let's move on to the next question. The plasma membrane lipid bilayer in cells has lipid arranged in what order? What can be the correct answer? Okay, any more answers? Okay, so the correct answer is option 2. Okay, I think the correct answer would be option 3 only, from head to tail. Okay, because this is how a arrangement should be in case of the lipid bilayer. So this is the head, and this is how it will be arranged. Okay, from head to tail. So it has a hydrophobic group, hydrophilic group, and the hydrophobic group, as we have discussed in the previous slide also. Okay, so which question do you need to be explained? The last one. Okay. Okay, so let's move on to the next question. Which of the following best describes the fluid mosaic model of the cell membrane? So what can be the correct answer? Yes. answers. Okay, so the correct answer is option number three, that is a dynamic arrangement of phospholipids and proteins that move laterally within the bilayer. Okay, so fluid mosaic model basically depicts that the cell membrane or the plasma membrane is basically a lipid bilayer that have hydrophilic heads outside and the hydrophobic tails inside. Okay, then it also describes the presence of the proteins that are present here. So there can be multiple types of proteins. That means they are transmembrane like this. So this is an integral protein or a globular protein. Then there are certain proteins that are linked to the outer surface of the membrane only, and some proteins that are linked to the inner surface only. Okay, so these are membrane associated proteins. Now we have certain proteins that are lipid linked to the lipid. Okay, so these proteins are linked to the lipid. So this is the outer cover, like one layer of the cell membrane, and this protein is linked to that particular layer, and similarly this one is linked to the other side. Then we have uh, proteins that are attached to the other proteins. So these proteins are basically attached to the integral membrane proteins. Okay, so this is basically the description of the proteins that are present in the cell membrane. Now there are no, I'm coming to that. Okay, so what happens is, if a cell membrane is basically living, okay, so that means this membrane or the phospholipid bilayer needs to be mobile, or it is fluid in nature. Okay, so that if we tell, like, what do we mean by fluid in nature is that if the cell grows, that means it is expandable because of the presence of this phospholipids. Okay, or the vice versa, if a cell has grown, then actually it can shrink to this particular normal size. Okay, so this was the first part. Second part is, there were certain experiments that were carried out, and it was mentioned that the phospholipids actually move from its positions. Okay, so and they actually can inter yes, so they can interchange between the layers. Okay, so that the interior layer phospholipid can actually move to the exterior one and vice versa. Okay, this helps in maintaining the uh, potential, basically redox potential of the cell. Okay, so this is how, one of the points is that they can actually move from one of the layers to the other. Okay, and the third point is to maintain the uh, homeostasis. Okay, these lipid layers, these cell membrane actually have a fluid nature. Okay, so if we talk about the temperature here, if the temperature is basically low or it's cold outside, so what happens to maintain the structural integrity and the components of the cell? What happens? This cell membrane actually becomes less fluid in nature. Okay, and if the temperature is high, so this becomes more fluid. Okay, so this is basically explained by the fluid mosaic model of the cell membrane. So, are the points clear or anything that needs to be repeated? Please let me know. Okay. Because this is an important concept that is basically variable in variable textbooks. Okay, so if no doubts, then we'll move on to the next question. Which type of molecule can pass through the cell membrane by simple diffusion? So, what can be the correct answer? Okay. Anyone answers? Okay, so please tell me what is polar means in terms of chemistry. Not really water loving. Yeah. So polar are those molecules that have charges. Okay. Water loving means hydrophobic. Oh, sorry, hydrophilic. Okay. Polar means that it would have a positive as well as a negative charge. Okay. Then ions are charged molecules. Okay. So if we have a plasma membrane like this, we have this is the head and these are the hydrophobic tails. So if we have a large polar molecule that have charges displayed, will it be able to pass through this membrane? Okay. So the first point, that is large polar molecules will not be able to pass through the membrane via simple diffusion. Okay. So first of all, what is simple diffusion? The basic definition. So simple diffusion is basically, okay. So here you guys are missing one thing. So it's right that we have to move a molecule from high concentration to low concentration, but the thing that is missing is a membrane. Okay, so that is basically we have a membrane here in case of the cell membrane. So how a molecule that have a high concentration outside will be able to move to a low concentration? So that means it would have to be small in nature. Okay, so okay, now moving on to ions. So ions are charged molecules. They can easily interact with the outer surface of the membrane, but they'll not be able to move to the inside because there is a hydrophobic gap in between. Okay, so that is why they'll not be able to move through this particular hydrophobic gap, and they will require certain proteins that are called as porine proteins. Okay, so or channel proteins, so that they can easily be shifted from the outside environment to the inside environment. Okay, so talking about the small non-polar molecules, so these small non-polar molecules can easily shift or easily come through the pores, come through the head, the hydrophilic head, and move through the hydrophobic region of the cell membrane and slide inside the cell membrane. Okay, so via simple diffusion, only small non-polar molecules can pass through the cell membrane. Okay, so the correct answer is option 3. And this is the explanation. So this is basically the extracellular fluid, and this is the cell membrane. So if the size is too small, so it can easily pass through the cell membrane. Okay, so all those hydrophobic molecules that are small in nature can easily pass through the membrane from their high concentration to the low concentration. Okay, so is the explanation clear? Any doubts? So ions are basically charged molecules. Okay. So what happens is, if will be a charge, then it will actually be it will be difficult to pass through this hydrophobic environment. That is the tail part. Okay, and this charge molecule is basically involved or interact with water. Okay, in many cases it interacts with water, so that's why what happens is it will not be able to interact with this hydrophobic environment. That would actually cause an energy shift. That's why ions are not directly transported via this simple diffusion method. Okay, very small amount, like in nanomolar amount, some of the ions can actually pass through, but that is not evident at all. Okay. So let's move on to the next question. Fluidity of the cell membrane decreases with. So what can be the correct answer? Okay. Any more answers? Okay. Any more answers? Okay. So, can you tell me the difference between the unsaturated and saturated fatty acids? Okay. What about unsaturated? Yes. So, not multiple bonds. How do we define saturated? Is saturated fatty acids are those fatty acids that have only single bonds between the carbon chains. Okay. Unsaturated fatty acids have, along with the single bonds, they have double or triple bonds. Okay, so this is called as unsaturated fatty acids. In case of the fluidity, if we see, so the fluidity of the cell membrane actually decreases when there is decrease in the amount of the unsaturated fatty acids. So here the correct answer is option 2. And how this is happens, so this is the saturated fatty acid that have single bonds in their chains between the carbons. And if there is any double or triple bond that is incorporated in between, then it is called as an unsaturated fatty acid. Okay. And what happens is, when there is a saturated fatty acid only in the phospholipid molecule, so it is represented as this, because there is no double bond in this particular case. And if there is any unsaturated fatty acid in the phospholipid, then it is represented by a kink. Okay, so that means it have a double bond or a triple bond in this case. Okay, so what happens if you guys arrange only saturated fatty acids in a lipid bilayer? So it will form a structure that is this kind of structure. Okay, so this is a structure that is rigid basically. So that have a specific shape. But if you actually incorporate a unsaturated fatty acid in this place, then it would actually lead to the presence of some space for the fatty acids to actually swim. Okay, so this kink nature of the fatty acids help the lipid bilayer to have some space for the lipids to actually have a swim. So it increases the fluidity of the membrane. So if there are more unsaturated fatty acids input in the membrane, that means the membrane is fluid in nature. And if there are less unsaturated fatty acids in the membrane, that means the fluid will be more rigid or less fluid in nature. Okay, so that means if the fluidity decreases, that means there is decrease in the level of the unsaturated fatty acids. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. So there are two cell membranes. So you are given with two cell membranes. One had 10 cholesterol molecules and the second one had 100 cholesterol molecules. Which of them will be more rigid? So what can be the correct answer? Any more answers? Okay. Okay. So the correct answer is option 2, that is the second membrane that have more cholesterol molecules will be more rigid. Okay. And here is the explanation. Okay. So there are different combinations of lipids that are present in a cell membrane. There is one more component that is cholesterol. So it is also a part of lipid molecule classification. So it belongs to steroid. Okay. So it also have a polar head group and a non-polar hydrocarbon chain, and this is a rigid planar steroid link. Okay. So, if this particular cholesterol molecule is present in a particular lipid bilayer molecule, so what happens is it actually stiff the region. Okay, so it makes the lipid bilayer more rigid because this region, this steroid ring region, is basically more rigid. Okay, so it does not help the lipid bilayer to move. So it makes them less fluid. Okay, so that means if a particular membrane has less number of cholesterol molecules will be more fluid as compared to the membrane that have high number of cholesterol molecules. So this will be less fluid. Okay, so any doubts in this question? Okay. Yes. Okay. Okay, so let's move on to the next one. If you walk through the mitochondria from one side to the other side, how many times you will see a lipid bilayer? Okay. Okay. How? So what you have answered, how will you see a lipid bilayer twice? And how once? Okay. So, okay, that can be considered it as an explanation. But this is a mitochondria. Yes. Okay. That is also another explanation. So, this is the outer membrane of the mitochondria, right? This is also a lipid bilayer, and then Yes. Now we have the inner membrane that is actually have some this kind of structure. So this is the inner membrane of the mitochondria. So you are here, right? Now you have to travel from one mitochondria to the other. Now you tell me how many lipid bilayers will you see? Okay, okay, okay. So first you will enter from here. Okay. So this was the first bilayer that you will see, right? Then you move to the inner membrane. That will be the second one. Now you have to travel through the mitochondria and you have to pass through this. So this will be the third one, and this would be the fourth lipid bilayer that you will see. Okay, like is the explanation clear? So we'll see the lipid bilayer four times. Okay, so the correct answer is option number two. Okay, so let's move on to the next question. How will you differentiate between a mitochondrial and a plasma membrane? So we have just now we discussed that mitochondria also has a lipid bilayer. Okay, plasma membrane also has a lipid bilayer. So how will we differentiate between them? Okay, any more answers? Okay, any more answers? Okay, so the correct answer is option one. So in the mitochondrial membrane, we have a specialized lipid called as cardiolipin. Okay, so here we can see. So as we know, there are different kinds of lipid that are present in the plasma membrane of the animal cells. We have glycerolipids, we have sphingolipids, we have sterols. Okay, so in case of the phospholipids, the base is the glycerol, and in case of sphingolipids, we have a base called as sphingol C. Okay, through glycerol, we have two fatty acids attached, and on that basis, we have a different specialized lipids called as phospholipids. Okay, then we have sphingolipids. In that case, we have sphingomyelin or glycosphingolipids in which like carbohydrates are attached. Okay. In case of phospholipids, we have different kinds like phosphatidylcholine. Then we have ethanolamine. Then we have phosphatidylserine. And the last one is phosphatidylinositol. Okay, so this glycerolipids or the general phospholipids are 65% in a cell membrane. Then we have sphingolipids. So these are sphingomyelin, and this is sphingolipids that are the base is sphingosine in this case. Okay, these are 10%. In case of brain, this amount is increased. Okay, so the brain cell membrane has high amount of sphingolipids. Then the last category of lipids is sterol. So this is cholesterol that is majorly present in the mammalian cell membrane, and this is 25% of all the lipids. Okay, now we'll move on to the mitochondrial lipids that are present. So they have phospholipids or the phosphoglycerolipids. Then we have cardiolipid. Okay, this is also have a glycerol as its base. Okay, so this is a specialized lipid that is present only in the mitochondria in the mammalian case, because it is basically derived from the bacterial origin. Okay, then we have phosphatidylserine and phosphatidylethanol that is basically a another type of glycerol lipids. Okay, so cardiolipin is a major category that is present only in mitochondria, and that we'll be able to differentiate between the mitochondrial and the plasma membrane. Okay, so any doubts in this question? Yes, Cl is basically cardiolipins. So it is only found in mitochondria, mitochondrial membrane basically.
So means it is found in mammalian cells also. Mitochondria
Yes.
layer of mammalian cells. Okay, sir.
Hmm. So any more doubts in this question? Okay. If not, then we'll move on to the next question. The type of lipids present in a plasma membrane are. Okay. Any more answers? Okay, any more answers? So yes, the correct answer is option 4. That means all these lipids are basically the part of the plasma membrane. Okay. So as I've explained before also, this is the phospholipid, this is a cholesterol, and this is a glycolipid. Okay. So all of them are part of the plasma membrane or the cell membrane. This is the major category that accompanies, so that acquires around 60% of the uh, composition. Around 10% is this, and the remaining 30% goes to the glycolipids. Okay, so this is how this is arranged in a plasma membrane. This composition varies from cell type to cell type, and it will also vary depending upon the fluid nature of the cell membrane. Okay, and this is another kind of lipids that are present called as glycerophospholipids or sphingolipids. So glycerophospholipids have a glycerol as a base. Okay, and sphingolipids has sphingofene as a base. Okay, so and in this case the head group actually is differentiated into different molecules, like there can be only phosphate, then phosphoinositol, phosphoglycerol, phosphoserine, phosphoethanolamine, or phosphocholine. So different lipid molecules have different functions. Okay, so phosphoglycerol basically helps in maintaining the integrity of the membrane. Okay, so if we talk about glycerol only, that maintains the integrity. If we have choline or inositol, that help in signaling. If we have serine, that is basically maintaining the negative charge on the membrane. Okay, serine as well as ethanolamine, both. So this is how different kinds of lipids have different uh, like functions in a cell membrane. Okay, so any doubts in this question? Okay, so yes, I was expecting this question. So basically what happens is whenever we talk about any cell, so it maintains a negative potential. Okay, so which means for any procedure or any signaling to happen, it need to have a negative potential. Okay, so this particular, if we talk only about the charge, keeping all the different parameters same, then a normal living cell, live cell is basically negatively charged. Okay, so this means that they are maintaining a negative potential so that all the procedures that they are performing are spontaneous or favorable energetically. Okay, so this is linked to the favorability in the energetics of the different procedures that are happening in the cell.
Sir, what is the initial charge of a living cell?
So it maintains only the negative charge.
Okay, sir.
Okay, so it doesn't have any charge previously, it maintains a negative charge for the proper functioning of a cell.
Okay, sir. Understood.
Okay, so any more doubts? Okay, if not, then we'll move on to the next question. During the formation of a cell membrane, what is the most energetically favorable step? So what can be the correct answer? Okay, any more answers? Okay, so you guys remember in the previous lecture, or yeah, in the previous lecture, I have told you guys that if the phospholipid bilayer is there, then it needs to be sealed. Does anyone remember why it needs to be sealed? So my question is why it needs to be sealed. Okay, actually it will not be termed as degradation. Okay, so what happens is, if we talk about a lipid bilayer, so we have a hydrophobic covering, hydrophilic covering, so that this hydrophobic region can actually live happily. Okay, so to maintain the hydrophobicity of this internal region, this membrane will be sealed. There will be no cut or entry space of water into this hydrophobic region. Okay, so is this part clear? So this is not be called as degradation. Okay, this would be in terms of favorable circumstances. That would be the best term that you can use. Okay, so what will happen if we have to find out what will be the most energetically favorable step in case of the plasma membrane formation? Then if we see, so the correct answer is option 2 basically, and here is the explanation. Okay, so this is a lipid bilayer. So this is a lipid bilayer. Okay, the pairing has already occurred. So this is the first layer and this is the second layer. And this white region is the hydrophobic tail of the fatty acids. Now, when this is actually exposed to a hydrophilic environment or water, so what will happen? It will try to curve itself and it will form a sealed compartment so that this hydrophobic region will actually get covered so that no water molecule interacts with itself. Okay, so in that case, if this we see, this is the pairing, this would be the most unfavorable step energetically. And as it has actually stabilized in form of a sealed compartment in the presence of water, then it would be the energetically favorable step. Okay, so if now the question changes, if this particular lipid bilayer is present in a non-polar solvent, will it form a sealed compartment like this? Because our body has around 70-75% water. Okay, so if the water is replaced by a non-polar solvent, then will this cell membrane will be able to form a sealed compartment? My question is that. Okay, how it will form a sealed compartment in a non-polar solvent? Okay, so what will happen with that? Okay, so the simplest explanation for that would be because here the hydrophilic heads would be outside that would interact with water. If the water is replaced by a non-polar solvent, then the reverse would happen. Okay, so what will happen? These hydrophobic groups will be inside and the hydrophobic tails would be protruding outside. So these will interact with the water, sorry, with the non-polar solvent, and these will be forming a pore so that it will have least interaction with the non-polar solvent. So is the explanation clear? Okay. So likewise, if we add water to this particular scenario where the hydrophilic heads actually interact with water and they form a sealed compartment so that this hydrophobic region is covered. If there would be a non-polar solvent in place of water, then the reverse would happen. Okay, this hydrophilic groups will try to save itself from the non-polar environment. Okay, and this hydrophobic tails would actually protrude outside, and that would try to form a region so that hydrophilic heads will have a least interaction with this non-polar solvent. Okay, now it's clear. Okay, so any more doubts in this question? Okay, so let's move on to the next one. Which of the following cells lack a cytoskeleton? So what can be the correct answer? Okay, any more answers? So please read the question carefully. The cells that lack a cytoskeleton. Okay. Hmm. So the correct answer is both the cells, either prokaryote or eukaryote, they will have a cytoskeleton. Okay. So to maintain the integrity of the cell, a cytoskeleton is needed, right? So both kind of cells, prokaryotes or eukaryotes, they will have the cytoskeleton. Okay, any doubts in this question? Okay, so let's move on to the next question. Which of the following is a function of a cytoskeleton? So what can be the correct answer? Okay, any more answers? Yeah, in this question we'll be doing that. Okay. So let's first answer this particular question, then I'll explain your doubts. Okay, so the correct answer is option 3, that is both A and B, that maintains the shape of the cells and it helps in positioning of the organelle in the cell. Okay, so what happens is, if we see, like, our body has a skeleton. Okay, so if we have to see, like, if we can stand, we can walk, we can actually like have a still body, so that means that is because of the skeleton. Okay, we can actually lift our hand, we can write, anything, that is because of the skeleton that is present inside our body. Otherwise we'll not be able to do anything, right? Likewise, the cell have also its skeleton. Okay, so the purpose of this skeleton, that is called a cytoskeleton, is that it helps in maintaining the shape. This circular shape or any different shape of any different cell is maintained by its cytoskeleton. Okay, then there are different organelles. Okay, how does a cell decide which organelle to be placed where in a cell? Okay, so this is also maintained by the cytoskeleton. So cytoskeleton have three components inside the cell. First is the microtubules, actin filaments, and an intermediate filaments. Okay, so microtubules helps in maintaining, helps in positioning of the particular organelle inside the cell. Okay, so if we see, this is a nucleus. Here are the endoplasmic reticulum. Here the Golgi is placed. So why Golgi is placed near the ER? Because it functions in accordance with the ER. Okay, so that is how it is maintained by the microtubules to basically help in the protein transport process, right? So likewise, any other organelle is maintained at a particular place because of the microtubules present inside the cell. Okay, actin filaments helps in maintaining the shape and integrity. Okay, so shape means that this circular shape. Okay, like we have different walls in our house. Okay, so walls basically helps in maintaining the shape of the house, right? So these actin filaments are actually maintaining the shape by actually linking or actually like stabilizing the cell membrane so that it will not fall apart. Okay, so this is how actin filament actually integrate maintains the integrity of the cell. Okay, so it will not it will help the cell membrane to maintain its shape so that the region or the organelle that is present inside the cell will not collapse, basically, like the walls in our house. Okay, then we have intermediate filaments. They also help in stability, and these are present basically in the nucleus. Okay, so this is how the major cytoskeleton in the cell works. So is this clear, the explanation? Okay, so let's move on to the next question. It is distributed in the whole cell. The cytoskeleton is actually distributed in the whole cell.
It means it is present in the membrane or where? Means in
Yeah, that's
cytoplasm
what.
is.
If you see, for example, I'm talking about the microtubule. Okay, so it will be distributed in the whole cell because if it has to maintain a Golgi apparatus here, then it will not only be present at the Golgi apparatus. Okay, so it will actually link so that the Golgi apparatus will be positioned in that place. Okay, so it will be like it will be present scattered in the whole cell. There is no specialized place for the cytoskeleton. It will be present distributed up in the whole cell.
Means it will mainly present in the cytoplasm.
Yeah, you can say that. That links the cell membrane also to the different organelle. Okay, so
Okay,
it
sir.
will be attached to the cell membrane. So any more doubts in this question? Okay, if not, then we'll move on to the next question. Dynamic instability is an inherent property of which of the following answers can be correct? Okay, any more answers? So the correct answer is option 1. It is only associated with the microtubules. Okay, so what happens is this is the case of the microtubule, and these are the monomers of the microtubule. So this is tubulin protein. So what happens in case of the polymerization? These tubulin proteins make the protofilaments. These single structures are the protofilaments, and during polymerization, different tubulin proteins attach upon itself. Okay. Now, when this depolymerization occurs, so what happens is this leads to the breakage and the removal of this dimer or the tubulin protein. So this happens as a routine process in a cell. So both rescue and catastrophe are termed as the instability in case of the microtubule. Okay. So in one case it would be growing microtubule because of the addition of the tubulin protein. In the other case it would be shrinking microtubule because of the decreasing tubulin proteins. Okay. Is the explanation clear? Mhm. Okay. So what happens is this microtubule actually helps in transport of the different molecules. Okay. So for example, this is a cell and you have to endocytose one vesicle. Okay. So all this transport process helps in with the help of the microtubules. Okay. So here the microtubule attaches this vesicle and it starts moving this vesicle from here to the lysosome. So this is a lysosome. Okay. So now what happens? How it moves? So basically, this is a chain of the tubulin proteins, and here is the vesicle. It will start destabilizing from here and start removing its tubulin protein from here. Okay, so that it will move the vesicle from here, then here, then here, and then to the end point. And likewise, it will add or polymerize the tubulin protein from here. Okay, so this will help in movement of the vesicle from the terminal, that is cell membrane, to the nuclear to the lysosomal membrane. Okay, so there would be one point where depolymerization would be happening, and there would be one point where polymerization would be happening. Okay, so is the explanation clear? Yes. Okay, so let's move on to the next question. So MS Dhoni, Rishabh Pant, and Sanju Samson, all of them are cricketers. All playing in one team is an example of which of the following processes that can also seen in cells. So what can be the correct answer? Okay. Okay. So the correct answer is option 2, that is redundancy. Okay, so what is actually common between these three is they are batsmen as well as wicket keeper, right? So if any one of them is removed or is not present in a team, then also the team can play, right? So redundancy is also the same thing. There are many components that have similar function. So if one of the component is absent, then they can actually like maintain the function of the cell, because the other components are present in the cell, and it will actually replace that particular component. Okay, so is the question clear? So this question basically explain us the redundancy in the genetic code. Okay, so for example, in the genetic code, we have certain codons that code for many amino acids, right? Or the other way around, many amino acids that are coded by more than one codon. Okay, so in that case, this is called as redundancy. So that if either of the codon is present, then also the amino acid can be coded, right? Okay, uh, just wait a minute. So, for example, let's take an example of phenylalanine. Okay, it is coded by two codons, that is UUU and UUC. So, if UUU would be present, then it will also code for phenylalanine, and UUC will also code for phenylalanine. Okay, so if either of them is present, then phenylalanine will be present. Okay, so that's how redundancy takes place in the case of cells. This is the best and simplest example that I can give. Okay, so let's move on to the next question. The microtubule wall is composed of globular proteins arranged in longitudinal rows called as. So, what can be the correct answer? Okay, any more answers? So, the correct answer is option 2, that is protofilaments. Okay, so what happens is this is a microtubule structure. Okay, it is maintained in a cylindrical way, and this one structure is called as a protofilament. This is what we are talking about. Okay, this protofilament is made up of polymer of a tubulin heterodimer. Okay, so this is called as a microtubule subunit. So microtubule subunit is a tubulin heterodimer. Microfilaments are present in case of actin. Okay, and this is a structure of the alpha and beta tubulin that makes the heterodimer. So these are arranged in the form of protofilaments, and this protofilaments then forms a microtubule. Okay, so this is how it looks. So any doubts in this question? Okay, if there is no doubt, then this was the last question for today. So thank you guys for joining in. If you have any doubt, you can ask.
Sir,
Okay. Yes.
sir, can you say what is nucleoporonin?
Nucleoporonin,
Poronin.
or it's nucleopore.
Yes, sir. Means from where the RNA is coming out from the nucleus
Okay.
and going to the cytoplasm.
Yes. So what happens is, so this is a nucleus basically. Okay, and there are certain pores inside the like on the membrane of the nucleus, that we call it as a nuclear pore complex. Okay, so that is called as a nuclear pore complex. Okay, so this nuclear pore complex has certain proteins that are called as nuclear porins or nucleoporins basically. Okay, so these are called as nucleoporins, where from this nuclear pore complex there are two pathways that follows. One, the molecules that goes inside the nucleus, and one from which the molecules from the nucleus comes outside to the cytoplasm. Okay, so this helps in basically transport of to and fro from the nucleus and to the nucleus. Okay,
Okay, sir. Understood.
okay. So any more doubts? Okay, if not, then we'll close the session for today. Thank you guys for joining in.